The human genome is of extraordinary complexity, with its 3 billions of DNA-based pairs that form the thousands of genes that make up our biology. Understanding this complexity requires powerful tools. Digital PCR is a powerful new tool for quantitative DNA measurements. Like real-time PCR, digital PCR uses a simple sample preparation that utilizes TaqMan primers and probes and an optimized supermix. Real-time PCR provides relative quantification. In contrast, digital PCR provides absolute quantification of target DNA sequences in the sample. Let's see how droplet digital PCR achieves precise DNA measurements. The key is sample partitioning. With droplet digital PCR, reaction mixes are partitioned into approximately 20,000 evenly sized droplets, each 1 nanoliter in volume. PCR is performed on each individual droplet. If a droplet contains the target sequence, it will be amplified and a TaqMan reporter dye will emit a fluorescent signal. If the target sequence is not present in a droplet, no PCR will take place and only weak residual fluorescence is emitted. By scoring each individual droplet for a fluorescent signal, droplet digital PCR achieves absolute quantification with a high level of precision and single-molecule sensitivity. To generate digital results, samples are amplified to endpoint in a conventional thermal cycler. Droplets that contain the target DNA sequence are read as positive. Those that do not are read as negative. The fraction of positive droplets determines the concentration of the target DNA sequence in the sample. Now let's look at how the Bio-Rad QX100 droplet digital PCR system performs digital PCR with unrivaled precision and sensitivity. The QX100 system uses a simple workflow that requires minimum hands-on time and procedures that are easy to perform. First, 20 microliters each of 8 prepared samples are transferred to the droplet generation cartridge. Droplet generation oil is then loaded into the corresponding channels of the cartridge. There are 8 oil channels, 1 for each of the samples. Once the cartridge is loaded with prepared samples and oil, it is placed in the QX100 droplet generator. Using microfluidic technology, the droplet generator combines the samples and oil to create an emulsion of approximately 20,000 mono-sized droplets for each sample. Every droplet contains a nanoliter volume of the sample, including any target sequences that may be present. After the droplets are generated, the emulsified samples are transferred to a standard 96-well PCR plate using a standard 8-channel pipette. The plate is then loaded onto a thermal cycler where PCR is performed to amplify the target DNA to endpoint. When PCR is complete, the plate is loaded onto the QX100 droplet reader. The QX100 droplet reader is an automated liquid handling device equipped with optimized microfluidics designed for precise, reproducible detection of target DNA sequences, droplet by droplet. The droplet reader automatically sips each sample, singulates all the droplets from a sample, and then streams them in a single file past a 2-color fluorescence detector. The detector reads each droplet in turn at the rate of 1,000 droplets per second and determines which droplets emit fluorescence and which do not. As each droplet is read, the data are recorded graphically by Quantisoft software. The software records the positive and negative droplets in each sample and plots the droplet-by-droplet fluorescence. This allows the data to be analyzed and visualized according to specific application and research requirements. The accuracy, sensitivity, and precision of droplet digital PCR enable a wide variety of applications that require accurate, absolute quantification. Examples of these applications include copy number variation, mutation detection, and gene expression. QX100 Droplet Digital PCR. The future of quantitative PCR is here, and it's digital. Be part of it. For more information about the QX100 Droplet Digital PCR system, visit our website or contact us directly by phone or email.